the zinc finger protein and first RNAs throughout the tetrahymena process.
Okay, so hi everyone, my name is Eva DeRango-Adem and I'm from the Perlman Lab at York University. And today I'm going to talk to you about some MAC-specific CHD proteins, CHD standing for chromodomain helicase DNA binding proteins, which is what Joshua Smith just talked about recently, right now. And we found that one of these CHD proteins binds a novel zinc finger protein through mass spectrometry, and RNA banding patterns, just preliminary research, have shown that they bind diverse RNAs, just qualitatively. So I'm not going to go through this very much because we had many rounds of this, but chromatin remodeling can occur in many different ways, and we're interested in the CHD family of proteins, which are ATP-dependent chromatin remodelers, which use the energy from ATP to physically manipulate the chromatin structure and allow it to be more or less accessible by other proteins. So the CHD family proteins are characterized by having two N-terminal chromodomains, followed by central SNF2 helicase ATPase region. And the CHD family proteins can be divided into three different subfamilies, depending on other accessory domains. The CHD family proteins in Tetrahymena include CHD3, which has a classical subfamily 2 domain architecture with two zinc finger domains in the N-terminal region of the protein, followed by the two classical chromodomains. However, there's another CHD protein, which is annotated as CHD1 in the Tetrahymena database. But this protein has a very unique domain architecture in that it has a chromo-bromo-chromo domain region in the N-terminal part of the protein, followed by the SNF2 central helicase region and 3 tandem PhD fingers. Now, previous analyses on using phylogenetic trees, I have identified from a previous graduate student that this chromo-bromo-chromo region in this CHD protein is unique to only aviolate species, including paramecium and many different parasites, including Cryptosporidium, Plasmodium, and Toxoplasma. And we've had some interest in Cryptosporidium as a potential drug target, so I'll talk more about a synthetic peptide that we've generated, which might be of interest for Cryptosporidium researchers, as this is a parasitic organism. So for the rest of the talk, I'll be referring to these two proteins as CHD3 and CHDAS, referring to as aviolate-specific CHD. So we don't really know that much about these CHD family proteins in Tetrahymena, and they have very interesting regulation and expression throughout the life cycle. So my research in the Perlman Lab is trying to identify different macromolecular interactors that these proteins bind, both through vegetative growth and conjugation, perhaps to elucidate their function. And the strategy that I'm using is something that I just called it for like a jazzy name, IP3, and this is protein immunoprecipitation, RNA immunoprecipitation, and chromatin immunoprecipitation, trying to identify these specific macromolecules throughout the life cycle. And I'll talk about one other functional analysis that I did using a peptide array. So IP3 is looking at three different macromolecules, DNA, RNA, and protein, and it relies on having a protein-tagged cell line with your protein of interest, in this case the CHD family proteins. Now, these CHD proteins are not known RNA binders, but some literature has suggested in high-throughput screens that CHDs can bind RNAs, in mammalian cells at least. So these are good candidate DNA and/or RNA binding proteins. So essentially, you have a tagged version of your protein of interest, and you pull down your protein of interest just by affinity purification, and then you either do an RNA immunoprecipitation or chromatin immunoprecipitation or protein immunoprecipitation and identify those macromolecules. So as I mentioned, CHDs are good candidate DNA and RNA binding proteins for a number of reasons. They are chromatin associated, and as you've heard from Joshua Smith, they are also involved in DNA damage as well as many other diverse roles. They are highly expressed in Tetrahymena during conjugation, but they are also expressed during vegetative growth and minimally during starvation. They have a unique domain architecture, and some of the domains, such as chromodomains in other model organisms such as Drosophila, it's been noted that these chromodomains can be RNA interacting modules. And as I mentioned, there are two main high throughput screens that have identified CHD proteins as mRNA binding specifically proteins. So we've generated FZZ tagged CHD proteins in our lab using the standard BKS FZZ vector that we have in 2 different mating types. And I did some developmental Western blots on these proteins to try to identify when, at the protein level, they were highly expressed. And CHD3 is highly expressed at the protein level at around 7 hours of conjugation, and CHD-AS, or the aviolate-specific CHD, at around 8 hours of conjugation. Following this, one of the great things about having this FZZ-tagged cell line, or any protein-tagged cell line, is that you can do indirect immunofluorescence using that same line. And this was done using confocal microscopy and indirect immunofluorescence against the FZZ tag. And like Alejandro mentioned with his protein, these proteins are MAC-specific, both during vegetative growth, starvation, and development. And there is a discrete switch from the parental macronucleus to the anlagen at around 8 hours of conjugation, and it's completely zygotically expressed. So following these preliminary studies on these proteins, I started with protein immunoprecipitation and trying to identify the protein interactors by mass spectrometry, which is done by a collaborator at Mount Sinai in Toronto. So CHD3 was one of the proteins that we identified in the preliminary screens. The research associate at our lab, Dr. Jody Garge, identified a novel zinc finger protein to bind the CHD3 family protein. And functionally, in the Tetrahymena genome database, homologs of this protein are E3 sumo ligase, so it's functionally interesting. I then followed up by doing a conjugation mass spectrometry or affinity purification of this protein, and again, mass spectrometry identified this zinc finger. So for the aviolate-specific CHD protein, unfortunately, no functionally interesting proteins were identified, but during conjugation, histone H2B was pulled down and identified by mass spec. So following identification of this zinc finger CHD3 interaction, the protein was reciprocally tagged, and the same analyses were done. And during conjugation, at the protein level, expression was highest at around 6 hours of conjugation. And again, that same phenotype of MAC-specific protein localization was seen in this protein. And mass spectrometry on this reciprocal tag identified CHD3 again, confirming this interaction. So just out of interest, we were thinking of looking at any potential RNA interactions with this protein. So RNA immunoprecipitation was done, and RNA immunoprecipitates were run on urea page. And unfortunately, no small RNA species were identified. However, and during vegetative growth and conjugation, there are similar RNA banding patterns. However, during vegetative growth, as shown by the purple arrows, there are 2 RNA species that seem to be unique to these 2 proteins. And during conjugation, there's only 1 RNA species that is unique, which is annotated by the blue arrow. So, of course, this is very preliminary data, and RNA sequencing needs to be done to qualitatively and quantitatively identify these exact RNA species. So finally, I'm just going to go into one of the functional analyses that I did, another functional analysis on this CHD-AS species, or CHD1, as annotated on the Tetrahymena genome database. And as I mentioned, it has a very unique domain architecture. It has that chromo-bromo-chromo domain region. And a while back, we had synthetic peptides generated for that region, as well as a tandem triplicate PhD finger, which is on the C-terminal part of the protein. And both of these proteins are well expressed. So the PhD finger wasn't really well expressed in this his tag vector, but upon expression in a GST vector, it's fine. So I initially did a protein peptide array using epigenetically modified histones, this commercial array that Alejandro spoke about, using this chromo-bromo-chromo synthetic peptide, which was purified in E. coli. So this is the commercial array. Again, it has about 394 epigenetically modified histones, ranging from acetylation, methylation, citrulation, phosphorylation, ubiquitin. There's many, many different modifications, some singly modified, some with multiple modifications on one spot. And this was the data that was created from the peptide array. Now, the 5 top hits that were identified in this array with the highest specificity, both in intensity and in replicates, was this H3K36 modification, both unmodified, monomethylated, dimethylated, trimethylated, and acetylated. So it's interesting that this chromo-bromo-chroma region with that unique domain architecture seems to bind these, this mark and specifically all of these different epigenetic modifications. So with any high throughput method, of course, you have to do some validation to make sure that it's not just a false positive. And we're considering many different types of validation methods. And if any of you have some advice for me, that would be very helpful. So that's the next step to validate this specific H3K36 CHD1 interaction. So I'd like to thank all of you and TAGC for inviting me to speak. Yeah, so that's all. Thank you. Questions?
Did you have a chance to look at the CHD the expression patterns at all?
At the protein level?
No, just of if there was multiple splice variants.
So I believe in paramecium there are actually multiple splice variants, but for Tetrahymena I don't think there are multiple splice variants, but I'm not entirely sure.
Well, we've been looking at it after DNA damage, and it does seem to have some of the introns at the beginning where they would splice out one of the bromo domain parts. So just was looking for validation too, and then it's on, and we could talk more later about that.
Okay. That's good.
So I was interested in your RNA IP. Have you those size those RNAs are sort of the size of some common structural RNAs like 5S, and have you tried just probing with probes, oligo probes specific for those structural RNAs just to see if that's what they are?
So there's 2 main bands that are present, and those are most likely ribosomal RNAs, as well as potentially a tRNA. So that's why RNA sequencing with ribosomal RNA depletion might be useful, because it was found in sometimes even wild-type pull-downs as a product of the tag. So that's definitely very necessary.
Okay, thanks.
So the mammalian version of this chromodomain binds to H3K4 trimethyl, but the yeast version I don't think does.
Vitro,
but I'm just curious.
On the array? Yeah, so on the array that site didn't come up. So I read that, I think in vitro, yeast didn't bind H3K4, but, and it didn't, also in this in vitro assay, it didn't bind, unfortunately.